right. Take a seat, take a seat. All right, I might get started. So I'm going to talk about advances in network neuroscience. I'm going to start with a really quick introduction to network neuroscience, where we are at the field now, and then I'm going to briefly tell you about some of the research that I have been doing. My name is Caio Seguin, by the way, at the University of Melbourne. All right. Cool. So network neuroscience and connectomics are fields concerned with the mapping and analyzing of brain networks. Brain networks can either describe structural or functional connectivity. So structural connectivity are anatomical connections between neural elements, whereas functional connectivity describes statistical associations between neural elements. So here we have the anatomical network of the C. elegans worm. This is a neuronal level network, where we have the complete synaptic map of connectivity between every neuron. And here we have a human brain network mapped from MRI. We can do this using diffusion MRI and white matter tractography. So this is a structural network. Or we can do it using functional connectivity. For example, correlation between fMRI bolt signals. So this is a functional network. Both networks can be represented either as this wiring diagram or an adjacency matrix. And the whole enterprise of network neuroscience and connectomics is to use tools, mathematical tools from graph theory and network science to try to make sense of this complicated sort of jumble of connections. People have been studying brain connectivity since the 80s, more or less, but in its present format, I would say that network neuroscience is give or take 2 decades old, with kind of early studies dating back from the early 2000s. So what have we learned in these 2 decades of network neuroscience? So we've learned that brain network architecture has a number of hallmark features. It has hubs, so highly connected nodes. It has rich clubs, so hubs tend to connect more preferentially with other hubs. It has modules of dense internal connectivity and sparse external connectivity. It has a small world organization that balances integration and segregation, etc. And these features are really universal. They are observed across species and spatial scales, all the way from the connectomes of C. elegans and going to the human connectome. So I would say that at least from the perspective of structural connectivity, there's a consensus that brain networks are organized according to this architecture. What there isn't a consensus about is whether this architecture really matters. So we might agree that brain networks are small worlds, but is that actually important or useful for understanding brain function? So another way to pose that is, are these properties merely descriptions of brain networks, or do they actually bear weight on mechanisms of brain function? And I think this is a really challenging question for network neuroscience to answer, but it's very important that we think about it. So the point of this symposium is to highlight research that's moving beyond mere descriptions of brain connectivity and towards models that are mechanistic, and if not mechanistic, at least phenomenologically predictive of brain function. We're going to hear from a variety of speakers, and we're going to cover how connectome architecture may shape very different cognitive, clinical, and physiological phenomena. Very important to highlight that I'm definitely not the first person to advocate for this shift towards predictions and mechanisms. There are a lot of papers that have done this before, and they have done this better than me, including work by Danny Bassett, Mika Rubinov, Jacob Vogel, who is speaking, and indeed Alessandro Gozzi. Okay, so now I'm going to tell you about 3 directions in my own research in which I've been trying to do this. So the first one is nanoscale network neuroscience. So whole brain nanoscale connectomes describe synaptic connectivity between every pair of neurons of an organism. So kind of the most famous and earliest whole brain nanoscale connectome that we had is the C. elegans one, and it had around 3,000 neurons. Then later last year, we got the larval Drosophila connectome with around the C. elegans had 300 neurons. The larval Drosophila connectome around 3,000 neurons. This paper is really cool. If you haven't heard it yet, it's amazing. And even more recently, we got a complete connectome of the adult Drosophila with 128,000 neurons. So there's a really rapid accelerating pace of connectome mapping, and also accelerating pace of the glamour of journals where these connectomes are published. I don't know where this one is going to end up. But the point here is that it's not about size per se. I don't think what matters here is that we have more and more neurons in our networks. To me, the really cool thing is that these nanoscale connectomes are richly annotated with neurobiological information. So we know a lot about each of these neurons, including cell function, cell type, morphology. For C. elegans, we know the birth times, we know about synapse polarity, and a lot more. And what's cool about this is that it gives us the opportunity to test whether these features of connectome architecture relate to the underlying neurobiology of these organisms. So I'm going to talk about a paper that I did together with Rick Betzel. And I really didn't really do it together with him. He did 99.99% of this. But here we are. So we looked at the larval Drosophila connectome. It looks like this. It has around 3,000 neurons. We get a nice adjacency matrix. And what we did is, based on this synaptic connectivity alone, we defined modules in this connectome. So largely, these are modules of dense internal connectivity and sparse external connectivity. And then we did some clever enrichment analysis to see whether these modules expressed cells with particular characteristics. And we find that they do. We find that these modules, which are computed on topology alone, delineate really well cell function, cell type, and cell group. So, yeah, this is what I just said. I think this is really cool because it's a step towards approximating a complex system view of the brain to a neurobiological view of the brain. Cool. So the next thing I want to talk about is prospective connectomics. And I'm going to explain what this means. And it's a line of research that, as far as I know, is only very recently possible as well, due to initiatives like the UK Biobank. So the UK Biobank has brain imaging for 40,000 participants in the UK, and it's the largest neuroimaging data set. And it also has very rich phenotypic data on these participants. But again, it's not necessarily about the size of the data sets. I don't think you need 40,000 participants to answer your research question. What is really cool here is that the UK Biobank has access to the health records of these participants, and these are updated through the platform. Of course, it's all anonymized. And what is cool is that I think for the first time, we'll be able to know what a person's brain looks like before they develop disease, or they present symptoms, or they have a brain injury or stroke or something. And I think this poses a really cool opportunity to study whether brain network architecture is predictive of these patient trajectories of resilience or vulnerability. Unfortunately, though, the UK Biobank does not have connectomes in the data that it shares. It has diffusion MRI, structural MRI, and functional MRI, but it did not map the connectomes into aerial connectomes. So this is what we did in this paper, working together with Sina Mansour, who is a neuroimaging wizard. I don't know how else to put it. We mapped connectomes for 40,000 UK Biobank participants, and this resource is now available to everyone. Well, you need to have a UK Biobank project, but it's available for download. So we have 28 out-of-the-box connectomes variations per subject. So these relate to different cortical postulations, subcortical postulations, pre-processing choices, etc. But Sina has also made some code that makes it really easy to generate even more variations of these connectomes, up to 1,000 different permutations of different pre-processing pipelines at a very, very small fraction of the time that it would take if you were actually downloading this data and doing this yourself. The code is all open source, and if you're familiar with the UK Biobank, these are now available as bulk files. I forgot to add Sina's GitHub, but there's a lot more information there. But if you just Google the paper, that will show up. Cool. And the last thing I want to talk about is brain network communication. This is sort of my primary interest. And the question is, how does connectome architecture shape communication between neural elements? So here we have a human connectome. We have two nodes, I and J, and they are not connected by a white matter tract here. And you might imagine that these two nodes are nonetheless functionally connected under some sort of behavioral paradigm. And the question becomes, how is it that the underlying structure of white matter connectivity facilitates communication or flow of information between these two regions? So here I'm highlighting a few different possible channels of communication. You have the shortest path between regions, you have a walk between regions. And these kind of reflect different conceptualizations of how we can model communication in brain networks. So yeah, it's kind of what I said. Can we use graph theory and network science to model neurosignaling? And these network communication models, what they try to do is quantify the structural capacity for communication under a certain assumption of how a signal might flow via connectivity. So the kind of modeling pipeline is that you start with your structural connectivity that is typically a sparse matrix. So it only encodes information between regions that are anatomically connected. And then you can simulate communication with a communication model, and that will give you a communication matrix that is now fully instantiated and tells us about the structural capacity for communication between every pair of regions. And what I want to stress here, this is very important, is that these models are super simple. This type of graph theoretical approach is not the only way to go about this problem. You can use dynamical models, you can use biophysical models, you can look at information theory. There's really a lot of different modeling domains to answer this question. And this one is perhaps the simplest one. It really abstracts away biophysical detail in favor of these simple conceptualizations of network communication. But I'm going to try to convince you that this level of abstraction actually buys you something really powerful. Okay, so we tried to see whether these simple models could actually predict the propagation of direct electrical stimulation in the human brain. So to do this, we used data for intracranial EEG in patients with drug-resistant epilepsy being prepared for resective surgery. They are implanted with these electrodes, and they can both record electrical activity at millisecond timescales and also deliver electrical pulses. So a limitation with this type of study is that usually you get very sparse spatial coverage because you only have data of where the electrodes are. And to kind of circumvent that, the FTRACT project compiled a lot of intracranial EEG data, so from more than 500 subjects, almost 30,000 stimulations, into this map, these two matrices of response probability and response amplitude, which kind of tell you across all of my sample, if I stimulate region A, what is the probability that I observe a response in region B? Or if I stimulate region A and I do observe a response in region B, what is the median amplitude of this response? So this is what I'm going to focus on, the amplitude. And by leveraging both inter and intra-patient variability in electrode position, they actually got a really good spatial coverage of the brain. So can we actually predict this signal propagation using simple models of network communication? And in particular, we were really interested in looking at propagation between anatomically unconnected regions. So for regions where you would truly need some sort of polysynaptic propagation through the white matter connectivity. And I'm going to focus on a simplified version of the results where I'm going to show you 3 models I'm going to very quickly explain the intuition behind these models. So the first one here on the left is the shortest path model. The shortest path model is really baked into a lot of measures of network neuroscience, often tacitly baked in. So if you compute local efficiency, global efficiency, characteristic path length, betweenness centrality, closeness centrality, small worldness index, all of these things assume that signals in the brain are flowing via the shortest path. So here we have 3 nodes, A, B, and C. These are the regions we're going to stimulate, and we have a target region T, and we want to see the effect, the response in T from stimulating these regions. So if the stimulus is going to flow via the shortest path, we would actually expect a stimulus to T to have a stronger response than a stimulus to B, then a stronger response in the stimulus to C. Because along the shortest path, A is only 1 hop away, B is 2 hops away, C is 3 hops away. Okay. Now we move on to this idea of search information. So in this case, if you notice, A, B, and C have the same shortest path to the targets. They're all 2 hops away in the network. What's different here is that the connectivity profile along the path is different. So the path starting from A is, filled with regions with lots of connections to the rest of the network, whereas the path starting from C has very few connections to the rest of the network. So this model measures diffusion of signal along the shortest path. So here we would predict that a stimulus to C would actually lead to the strongest response because the stimulus has less opportunity to dilute and diffuse to the rest of the network. And if we take this idea of diffusion and broadcasting to kind of its extreme, we get to the model of communicability, which tells you that every single route between 2 regions actually carries weight of the signal propagation, actually matters. So here, A to T has a shortest path of 2, B is the same as shortest path of 2, but if you stimulate A, you have many short routes to your target, whereas if you stimulate B, you have these long routes to the target. So the idea is that a stimulus to A now would lead to a stronger response than B. So what did we find? This is what we found. We found that so search information, SI, and CMY communicability, not only they could predict the amplitude of signal propagation, they could explain significant variance in that, but they did it a lot better than the mainstay measure of shortest paths. So this is saying that kind of this diffusion conceptualization might be more explanatory than a shortest path conceptualization. And just to be clear, this is signal propagation via a causal intervention traveling at millisecond resolution between anatomically unconnected regions. And we can use these really simple models that are analytical transformations of your structural connectivity matrix, and we can predict that with pretty good correlations. Okay, but we know there's problems with connectome mapping from diffusion MRI. Electrical stimulation travels via volume conduction. The Euclidean distances between regions is a huge confound. It's data from epilepsy patients, which adds another confound. So can we do the same thing, but instead predicting the propagation of single neuron optogenetic stimulation in C. elegans? So there's this paper by Randy and colleagues out last year in Nature, where they did, this is just mind-blowing. So they built this rig where you put a worm in there, you can optogenetically target any neuron you want while recording calcium imaging of the whole system. And it's all automated, it's amazing. This paper is fantastic. So we got in touch with these guys and we've been working with them to try to see, oh, sorry, I forgot to mention, and after you do all that, you get this very similar to the F-Track project in humans, this causal stimulus propagation matrix, where if you stimulate one neuron, what is the amplitude of the response in another neuron? So we got together with these guys and we asked, okay, can we use the same models now computed on the C. elegans connectome, which is a bona fide connectome where we actually know how everything is connected, to predict this stimulus propagation? And it turns out we can. Which is, I don't know, this result is just last week, but I'm so happy about that. I didn't know if it was going to work. So this, again, is stimulus propagation between anatomically unconnected neurons in this case. And what is really cool is that I think this really exemplifies the power of simple models. So we have electrical stimulation in white matter connectivity. We have optogenetic stimulation in synaptic connectivity. And we can use exactly the same models to predict both things. These models are not mechanistic because they are very simple, but they give a really nice interpretation of how these architectural features of a network can shape stimulus propagation between regions. I think it would be really hard to use the same biophysical model here to model stimulus propagation across these two species. So I think this is a real strength of these simple models. Cool, so this is my last slide. Other advances in network neuroscience. So we're going to hear from Jacob Vogel about models of neurodegeneration spread. We're going to hear about, from Ariana Menardi on network control theory. So if you're not interested in how a signal flows from one region to the other, but you're more interested in how a stimulus might change the overall state of your system driving that, that's what you're going to hear from. And we're going to hear about connectome dynamics in multiple time scales by Sepide Sadagiani. And just very quickly, other lines of research that are really interesting, but I couldn't fit everyone in the symposium: connectome generative models, neuromorphic neural networks, and biological annotations of human brain networks is also really cool. So that's it. That's all I have. We're going to save the questions for the end, and we're going to do a panel style, but thank you very much for the attention.